Hi, dear students. Today we're going to talk about a very important as well as an interesting topic, the journey to DNA. DNA, the term DNA, is quite familiar to every one of us. We find DNA's applications in the identification of COVID patients. We use DNA to identify criminals. The paternity tests are being carried out using DNA. You have touched DNA. Like that, DNA is something very much common today to every one of us. But there was a time at which DNA was totally unknown to everyone, and it is a work of various scientists uh, which led us to the information that DNA is a genetic material of man. So today we are going to go through a series of animations as well as some informations through the experiments of various scientists who led to the journey of the true DNA which we have today. So let's go into that. The journey to DNA. The first piece of this puzzle mainly involves the discovery of DNA by Frederick Misha. Coming to, if you read about him, you can see that his work is something very much important. In 1869, it was a landmark year in the genetic research because this was a year at which this Swiss physiological chemist Frederick Misha first identified what he called the nuclein inside the nuclei of WBC cells. He started with the aim of identifying protein, but as a part of serendipity, he happened to identify something which was of different nature, which was being found to be rich in phosphorus and which was resistant to proteolysis, and he named it as nuclein. Fred Misha, he wrote that it seems probable to me that a whole family of such slightly varying phosphorus-containing substances will appear as a group of nucleins equivalent to proteins. That's what he wrote at that particular time. Now, today I would like to, rather than saying what he did, I would like to take you through an animated video which was being put forward by the DNA Learning Center, Cold Spring Harbor Laboratory, with their animation showing how the hero of this experiment, Frederick Misha, led to the discovery of DNA. Now let's explore the site, which is dnaftb.org by 15. DNA from the beginning, which is a website which has been put forward by the DNA Learning Center, Cold Spring Harbor Laboratory, and in that they have laid down a concept called DNA and proteins as a key molecules of cell nucleus. So you can just go through their animations, and here we go to next our person of interest, that is Frederick Misha. Let's see what is it in this animation. This is Frederick Misha, as we are seeing, the man who discovered DNA, and his writing is just like an autobiography and animation is written. I was interested in chemistry of cells. In 1869, I began working with white blood cells. And white blood cells are the major component of pus from infections. I collected a lot of pus from the bandages at a local hospital. This is the bandages that he took from the local hospital. And I used a salt solution to wash the pus off the bandages. When I added a weak alkaline solution to the cells lysed and the nuclei precipitated out of the solution. So he's putting the bandages in salt solution He's putting an alkaline solution into it and something got precipitated. That was the cell nucleus, which has been found to be at the bottom. From the cell nuclei, I isolated a unique chemical substance which I called nuclein. I found nuclein in every cell type tested. Chemically, nuclein is high in phosphorus. Therefore, I thought nuclein mainly functioned as a cellular storehouse for the phosphorus atom. In my lifetime, we were not able to show any other clear biological function of nuclein. Now comes the next scientist who played an important role along with Frederick Misha. This was Phoebus Levin. And by the early 1900s, we knew that Misha's nuclein was a mix of protein and nucleic acids. There are two kinds of nucleic acids. DNA is found mainly in the nucleus. RNA is found in the cytoplasm. And although most of the work was done on the chemical nature of DNA, let's first talk of the proteins. That's what is being related to Phoebus Levin's work. Now, protein is made up of 20 different subunits, amino acids, and these amino acids are chemically linked together to form the protein chains. And this is a hypothetical protein chain. And with 20 different amino acids to build from, even a small protein can have millions of sequence combinations. Let's take an example where we link 4 amino acids. Since any of the 20 amino acids can occur at each position, there are 160,000 different possible combinations which is possible using the amino acids. Now, if you put the lever here, you can see the different amino acids which is possible with it. Therefore, proteins come into different sizes and shapes, and Levin believed that variety of proteins are a good candidate as a molecules of heredity. Those days, the concept of nucleic acids or DNA were not evident, and Levin thought that it is a protein chain that was responsible for the heredity rather than a DNA. DNA molecule that I worked on has only 4 different units. The I here is Levin. He worked on 4 nucleotides. What is a nucleotide? It is made up of 3 elements. We have a phosphate, you have a deoxyribose sugar, as well as a nitrogenous base. And the carbons of the deoxyribose sugars are numbered 1 to 5, and in the nucleotide, the nitrogenous base is always bound to the carbon 1. A hydroxyl group is bound to the 3, and the phosphate is bound to the carbon 5. This is the cytosine. In the next one, adenine was linked. Then you have another nucleotide which thymine was being linked. And then you have another nucleotide in which guanine was linked. So, Levin figured out that nucleotides are linked by a phosphodiester bond. This is a phosphodiester bond. And he thought that nucleotides were found in equal amounts in the DNA molecules. And he proposed that DNA must be a tetranucleotide, a linked series of 4 where the order was always the same. In 1938, he found that DNA was a much larger molecule than anyone had thought, and it made more sense to think DNA as a polymer and a long chain molecule made up of repeating units of tetranucleotide. It was hard to see how DNA could be intelligent enough to carry out heredity information. In his model, DNA is made up of tetranucleotide blocks, whereas proteins have an alphabet of about 20 amino acids. So in his opinion, proteins were a better choice as a nuclear hereditary material rather than DNA, because he knew only about the tetranucleotide DNA chain. Now, it is a work of these two scientists which led us through the various other expansions and enlightenments in the DNA field. First, we had Misha, and then we had Phoebus-Levin's experiments, which also felt us think that DNA was not the genetic material, but it was the protein which was a genetic material. So we will be continuing in the next series, the next experiments which led us to determine that DNA was a genetic material and not the protein. So thank you for now. Hope so you enjoyed this animation with the help of the lab Cold Spring Harbor Laboratory. Thank you for now.